This short lecture will introduce you to the techniques used to isolate bacteria from a mixture or ensure that you have a pure culture of your bacteria of interest. In nature, bacteria occur as mixed cultures. Swab your skin, swab your keyboard or cell phone, isolate bacteria from a plant, from your dog's mouth, and there will nearly always be more than one kind of bacteria. And to study the characteristics of a single species, it must be separated from the mixture. So how do you obtain a pure culture? There are a couple of ways to isolate individual bacteria from a mixture in order to obtain a pure culture. In either method, you're essentially diluting the bacteria in order to achieve single isolated colonies. And once you have the isolated colony, you can pick that colony, transfer it to a new sterile medium, either a plate or broth, and allow it to grow. One method used to obtain isolated colonies is the quadrant streak method. You spread your bacterial mixture onto 1/4 of the plate. You can see that in the top smear on the plate or under A on the drawing of a plate. Then you flame your loop to eliminate the bacteria on the loop. Then taking your loop and starting where you first streak the bacteria, you streak some of the bacteria into another quadrant of the plate. You flame the loop again, and now placing your loop in the second quadrant, you streak bacteria into the third quadrant. And then repeat until you have streaked bacteria onto all 4 quadrants of the plate. In the final quadrant, you should have diluted the bacteria sufficiently to produce isolated colonies. And you can see that in the photo on the left of a plate of bacteria. In fact, when maintaining bacterial colonies, we typically make quadrant streaks because it's always desirable to have isolated colonies. In case there should be some contamination on that plate, you can still go in and pick one of those isolated colonies that's free from contamination to start a new plate. In the quadrant streak video, I demonstrate how to do the quadrant streak. Another method is to begin with the broth culture and make serial dilutions of the bacteria. By doing a serial dilution, you dilute the number of bacteria in each dilution. In the drawing, you can see that with each dilution, the bacteria is diluted 100-fold. In this case, we're putting 50 microliters of that bacteria suspension into 5 mils of broth, so diluting it 100 times. And in this exercise, which we'll do later in Biology 207, you spread mixture from the last 3 dilutions. In this case, the 10 to the -6, 10 to the -8, and 10 to the -10th dilutions. You spread about 100 microliters of that bacterial suspension onto agar plates and wait for growth, and you should have isolated colonies. We don't use suspensions from those first few steps in the dilution series because the bacteria is still too concentrated. So these are 2 easy methods commonly used to grow isolated bacteria. And you will use both of these techniques in the Labster simulations.